a month kind of thing, so I have to all right, just proceed. I will turn off my video during the lecture or Amali because there might be some interruptions. Okay, today Amali will be covering, as I've mentioned to a few of those who came in a bit earlier. Can you all hear me? Boleh kan?
Boleh, Doktor.
Doktor.
Boleh, Doktor.
Today's practical will be sort of a briefing for what you will do throughout your practical sessions later on. So necropsy, all right, and how to post-mortem the fish, as well as fresh smear and a little bit on blood sampling. Blood sampling is not crucial because usually we use it for research, most of it for research. It's very rare that we need to take blood and run, you know, all the hematological parameters, very rarely, but I will just show you how it is done. Last week, Friday, Prof. Sharif talked about ectoparasites and how a bit on how to do fresh smear, or we call it wet sampling. So he mentioned that it is to detect or confirm ectoparasites and also fungus. So we'll cover that today a little bit. Well, not a little bit, it's a sort of a briefing, so you guys will know what to do in your coming practical session, which starts on the 29th. Okay, I hope that you guys keep this in mind. Actually, some of the pictures here are from the World Fish sampling guide that I've put in Padlet. So when you want to come for the practical session, please bring this or that handout so you guys can have something to refer to besides my practical handout that I will give. So the practical handout by the sampling guide by Worldfish is more of a graphic, a lot of illustrations. So I suppose it's more easier to follow, as opposed to mine would be sort of instructions rather than a sampling guide. All right. So you guys need two of those when you come for practical session, the related sampling guide and also my instructions or handouts. Okay, I hope that is clear. Okay, I will start. I hope most of you guys are in. Okay, I think, sorry. Farha, can you give them the link of Google Form attendance?
Oh, yeah, Doktor. I already did.
Alright, just paste on the chat box as well. Okay, I will record. I always forget to record. I'm so sorry, but I will start my recording. Okay. Okay, for ectoparasites and fungus, what we will do is a wet mount sampling, all right? Or we call it fresh smear, right? So most of the previous or old, not old lah, I mean like old, not older lah, but you know, Prof. Sharif call it fresh smear, okay? So the right, sorry, a lot of things are coming up. All right, the reason or the purpose of doing fresh smear or wet mount sampling, all right, is to take samples on the skin and as well on the gills, okay? So for the mucus sample on both sides of the body, all right, we do it by skin scraping, okay? And for the gill samples, we take tips of the gills, okay? For the for the wet mount sampling, all right, as I've mentioned before, ectoparasites and fungus, first thing to do always is to label your slides, all right? Okay, label your slides according to the date and the sample type, right? And if you have a fish ID or a farm ID, then you would want to put that as well. Okay. Next is when the fish comes, it will usually come, we will bring it in a few tanks, all right, or aquarium or aquaria, all right. What you would want to do is we will provide you with a glass beaker. So you kind of like take a few mils, 10, 20 mils of water from the aquaria and then put it at the side on your table. And apa tu, you can drop, alright, take 2 to 4 drops of water to put on your glass slide, alright, and place the euthanized fish on a clean surface. Usually we will provide you with a tray and you put a few wet towels, alright, wet paper towels before you put your fish on the tray. For practical sessions, this earlier part, we would not recommend to euthanize the fish with chemicals, but rather to do pithing, all right? So I forgot to put in how to do pithing here. Perhaps we can look at a few videos later on. So you use a very sharp scalpel blade, all right? You place it in between the head region and the slightly before the dorsal, well, not dorsal fin, slightly before, let me just show you. Right, you place your blade here, right, between the head and the body, right? You will find a soft spot where you can place your blade into, so you cut through, not it's to decapitate, but not wholly until the head comes out, right? So once you reach the spinal cord, you take a very sharp, apa tu? Very sharp, what is it called? Not needle, needle also you can use, alright, needle, and you kind of like scramble the the brain. So that's how it works. So the fish will not be able to feel anymore after that, but it's alive, but to a certain extent. All right, so you will be able to do your sampling meanwhile. Okay, so it is a humane way of killing fish, and we do it for our practical session if we are not using a chemicals. We can use chemicals first to sedate and then do decapitation and pithing, but I would rather that you just pith the fish immediately so that it saves a lot of time. Okay. Um, okay. So for skin mucus or lesion or ulcer sampling, okay, so you take a sharp, you use the back, all right, the back of your scalpel blade, and then you go along the body from anterior to posterior or dorsal to ventral, all right, so you will get a mucus and a little bit of epithelial cells. Try to avoid taking too much of the scale, okay? So you would do that behind the pectoral fin and along the belly. So you go anterior to posterior, all right? And oops, and then you place it onto the glass slide, all right? You can just rub it at the side of the glass slide, or you can use another maybe forceps or another blade to put the mucus on the glass slide. And then don't forget to add a few drops of water, not tap water, okay? Please remember, do not use tap water because it will cause any ectoparasites to shrink and die, you know, because they also have a membrane, all right, cell membrane. So when you put in tap water, right, osmosis happens and then the parasites can die, okay? And then once you've done that, you put in your cover slip and then you can view the sample under the microscope. Similarly, for the lesion or ulcer, you scrape the infected area and then you perform the same steps, all right, until we can view it under the microscope. Okay. The thing that you would use for gill sampling, all right, so that's done. That is to view or to take sample, mucus sample from the body, to check for ectoparasites. Another area that you would want to check for ectoparasites, as Prof. Sharif has shown last week, is the gill area. Okay, so you use a fine tip scissors. All right, you take at the just very tip of the lamella, right? And then drop the gill clippings onto the glass slide, and again do not forget to add a few drops of water, and then try to spread them out with your forceps. All right, so you can view the individual secondary filaments or primary filaments if possible. All right, and then place on your cover slips. Alright, so again, I would want to remind you to remember to add in a few drops of water to the edge of cover slips to prevent drying. If it dries and then when you put it under the microscope, the light microscope, with the lighting from the microscope, it will dry even faster. So at the end, you will not be able to view whatever parasite that is under your microscope or on your glass slide, right? So the technique is to go kind of like this type of movement, right? This loop of movement to cover all areas of your glass slide or your sample. All right. Move the field of vision in a continuous S-shaped movement to cover the entire slide. For skin and fin scraping, all right, what can be seen? Usually, sometimes in healthy fish, we can see a little bit of protozoa, but it's fine, all right. Sometimes one or two, that is totally normal, but you would want to take into consideration if the fish has clinical signs and also the number or the burden of the parasites on the samples that you have seen. So if you see a heavy burden of protozoa, then definitely, you know, something is going on and you need to check, all right? And certainly when you take lesion or even you're looking at the gills, sorry, this should be skin, fin and gills, okay? You can see hemorrhage, erosion and ulceration, right? Hemorrhage and erosion, ulceration can be on the skin. For the gills, you can see hemorrhage and hyperplastic cells. Okay. All right. For gill biopsies, certainly if you find a heavy burden of protozoa, you will be able to see hyperplasia, hypertrophy of the epithelial cells, and fusion of the lamella, telangiectasis or dilatation of the small blood vessels, all right, could be due to toxin or improper biopsy techniques. And if you have a bacterial infection, all right, this will damage your gill tissues and cause necrosis. All right, so how does it look like? I have some pictures, but Dr. Anas will be able to guide you better, all right? So if you see you take your skin or your gill sample, all right, usually commonly we would be able, in a heavy burden now, right, in a sick fish, you'll be able to see Ectopteris multifilis, right? So Prof. Sharif mentioned before, it's a round shape with cilia around it and a very prominent horseshoe shaped nucleus, right? So this is just an example. You won't be able to see on each and every sample that you take, all right? If the fish are healthy, then it is a little bit difficult and we'll talk about that later, all right? Salmonella, alright. Sharif has mentioned this before. Trichodina. Gyrodactylus or gill fluke, you can see not only on the gills, sometimes you can also find it on the body, alright? So remember some of the structures that Prof. Sharis had mentioned before, okay? The hook, right? So it's very prominent or very easy to identify, right? And for the crustaceans, you can get Lenia. Alright, Lenia, this one is selalunya it's visible by the naked eye. So if you would want to take one and then view under microscope, this is how it would look like. Okay. And do not forget your this is not lice. This is a copepods, alright? Copepods that you can this is actually my experimental fish. Right, so I decided one day after receiving the fish, I just want to check the fish, their health, and then we found two in this fish. So obviously if it's my fish, then it's from our group lah kan. So it's kind of like disgusting, you know, when you look at it, it's like you macam tak boleh nak makan lah bila tengok benda ni. Macam so it's like a cockroach kan but in your mouth. So yeah. Okay, for blood sampling, usually we do it for research purposes. Okay, there are a few ways that you can do it. You will learn in your rotations. Okay, but basically, you would need to determine the needle size, syringe size, as well as the location. Okay, so for fish, um, you can do it either the fish will be alive later or you want to kill the fish to take the blood. So for that, you can use a hematocrit glass capillary tube. Okay, you kind of like sever the fish in half and then you take the blood. The usual vein that we take is right under the vertebrae. So the bigger veins lies under the vertebrae. So you cut the fish in half and then you run your capillary tube just under the vertebrae to take or to need the blood, alright? For juvenile fish, alright, you can use 1 mil 30 gauge, or usually I use 25 gauge, right? Sometimes, especially for marine fish, you would need an anticoagulant to help you take blood, right? Because as soon as the blood enters the syringe, it will clot, especially for marine fish. So that anticoagulant, we can actually purchase it, and then we take the anticoagulant into the syringe and then kind of like coat the syringe before we take blood. So, but this is not so much done for disease diagnosis, right? Right. So needle and syringe with anticoagulants, heparin. I've only heard of. I mean, one example is heparin. There are a few other examples, right? So it's caudal vessels. We need to sedate the fish. Needle at skin near the base of the caudal peduncle, right? 3 scales down from the lateral line. For other methods we can use is via the heart. So you poke your needle in through where the heart is or the pericardial cavity, or you can sever the tail and use the hematocrit tube to take blood from the caudal vessels. Alright, so this is an example, video examples that you can see. Right. Alright, before we go to necropsy, I think I want to show you how we do beefing. Alright, so I will stop. Am I sharing my content? Okay, hold on. I will try to see where I can find the video. Let's see, there's a good video. Tak adalah, but anyways, I'll show you a photo instead. Alright, so when you do your practical session, okay, so this is the head region and then this is the body region, okay, and so you go in with your apa tu, scalpel blade, right? You go in here until you kind of reach or you kind of hit the vertebrae, or you can agak-agak with when you look at the lateral line, okay? And then put in your needle right towards the brain area, okay? And then kind of like spike the area in a round motion, right, with the needle, okay? So that would be able to injure or is not destroy lah, but be able to prevent the fish from feeling pain afterwards. Okay, I hope that is understandable. Okay, so that is what you will do for your practical session. Okay, I'll continue with my slides. Okay. Are you guys in? You're very quiet today. I feel like I'm talking to myself.
Ada je doktor.
Ada doktor,
Okay, okay. I
we're
hope
in.
can you all understand what I've said before, right? Especially the pithing
Faham?
Yes,
method. Right.
Doktor
Great.
kan?
Because you guys will do that most of the time in your practical session. Okay. I've talked to the staff. What we will do is we will lay out everything on your table. All right, including the fish. So whatever fish that you will need will be on your table instead of you having to go to the side left or right to take your fish. You know, it'll be very hectic. So everything will be on your table, your glass beaker, your tray, your paper towel, your garbage bin, and everything. You need to bring your own dissecting set. Okay, make sure that you have in at least not if not new a sharp scalpel blade. Very important. Ada dua size kan. At least have both lah. Okay, sometimes if the fish is small, like a small koi, right? So yang besar, if you use the bigger size, you wow definitely terus decapitate the fish. So you wouldn't want that. So you have to use the smaller blade. Okay. So I mentioned before, you be able to cut in between the head and the trunk region, right? Kind of like pierce the brain, so that the fish won't be able to feel. Okay, just up to refresh you guys on the fish anatomy. Alright, gills, right? So this will be heart, right? This will be the area if you want to take blood from the heart. If you do so, the fish will not be able to survive afterwards because the volume of blood in the fish is unlike mammals. Alright, so if you take 0.5 mil, that's already going to kill the fish. It depends on the size, but for a juvenile fish, that will kill the fish. If for a bigger size fish, 1 to 2 mil kill the fish. Alright. So when you want to go for your practical session also, if you are having trouble to determine which organ is which or where, you can bring your own, or you can just Google during the class, or bring a printed sheet of the fish anatomy. So you would able to help, you would be able to help yourself and your friends to determine where the organ is. Because although it is similar, when you open, it'll be quite small, it'll be a bit difficult for you to see. So having a guide will be able to help you. All right. So some fish will have some fish, as I mentioned before, the anatomy will be more or less the same. Others will not be the same. Like we will provide you guys with claria or ikan keli. So if you have time, you want to be able to look at the accessory breathing organs, right? And look at how the intestinal anatomy is different from other fish, if they are different. Okay. All right. For necropsy. Okay, so basically, what we do with necropsy is after you have done taking your fresh smear, taking your gill sample, and taking your blood. Okay, if you want to take blood, okay, we will proceed with necropsy. Okay, this is also for endoparasites and also taking sample for bacteria and also histopathology samples. Okay. So as with any other necropsy, you would want to maintain sterility. So if possible, especially if you want to take sample for bacteria, kan? So you want to be or to remain as sterile as possible. Okay, where to start? There are a few ways, but generally, it depends. All right, there are two main ways to do it. Some people do it one way, different people do it another way. Depends. Sometimes it also depends on whether you're left or right-handed. Okay, so whatever it is, think about how to maintain sterility. And if you are comfortable with the technique, or if it is easier for you, then use it. All right. So you can start from the head, go posterior, all right, or you can start posterior just in front of the urogenital or the anus opening. So you go anterior, okay. For the first method, all right, you cut near the or in the isthmus region, all right, isthmus region, and then you go caudal or posterior, all right. For the other method, you make a nick with a scissor. So you nick, right? And then you put in your scissor with sharp tip and then go. This is the head, right? So you go to the head region, all right, until you reach the isthmus region, and then you want to go dorsal, okay? All right, so if you start from the anal or the urogenital opening, so you make a nick and then you cut with a scalpel blade. Usually I just use a scissor. Very easy to use scissor because you can avoid this damaging the internal organs. So you go anterior, right, or dorsal, and then you go anterior and then you go dorsal. All right. You cut. All right. Just in front of the pectoral fin, right? Just before the operculum, and then you go dorsal. The peritoneal or the peritoneum, all right, wall will be able to guide you when to stop, all right? It's around where the lateral line is. Okay, and then you go caudal, and then meet the line at which you cut. Be careful not to cut through the urogenital opening because then you would expose the intestinal content to the sterile organs, right? So the other way is to start at isthmus, all right, and then you move caudal, all right, along the body wall, right, and then you go anterior or dorsal, and then you go anterior, all right. So the body abdominal cavity wall will be a guide for you to cut without damaging the organs, right? And then you can remove this flap lah. Usually the pectoral fin is in that flap, all right. Now, some things to keep in mind when you're doing necropsy is to remember to keep the fish moist with a wet paper towel. Okay, so we provide you with paper towel. You can dip that towel in the tank where the fish is, or you can take a beaker and then just, you know, bathe the fish with not bathe the fish, bathe the paper towel underneath the fish with the aquarium water. Alright, once open, observe the cavity and organs for changes if there are any without manipulation. So without touching the organ first. So you look at it. How does it look like? How does the peritoneal cavity looks like? If there is any endoparasites, you will be able to see it's a very fine white thread. Macam very fine. It takes practice to see, but once you find it, you will be able to do it the next time, all right? Sometimes if they are gravid, all right, especially the female, you can remove the gonads if necessary because it will usually cover 50% of the abdominal cavity, which will hinder you from looking at the internal organs. So you can remove that. Right, so you start examining your internal organs, the fish internal organs systematically, okay? So you start from the pericardial cavity, the heart, right? And then you move on to the digestive, liver, gallbladder, spleen, and then gonads. And don't forget to look for air bladder also, right? The gas bladder or the air bladder. Sometimes it can be congested as well if there is any infection. Right, once you're done, you can pop the bladder, all right, if you're done looking at it, to reveal your kidney, okay? So the kidney usually is where we take samples for bacteria, but virus, usually is the kidney, okay? Not liver, kidney. Usually lah, it's always kidney. Anything systemic, fish, kidney, okay? And then you can, if you have time, not necessarily, you can look at the cranial, alright, cavity, or you can open the cranial, alright, cranium, okay, and to reveal your brain, okay, not your brain, the fish brain. With a smaller fish, the bone is thin, so you can use you can use scissors to open. Right, so the brain is like this, right? So the cranium is covering it. So you cut left and right and then open the cranium to reveal the brain. Okay. In smaller fish, easy. But if too small, all right, maybe a fish this size, all right, 2 to 4 cm, it's a little bit hard. But if you're determined enough, you can do it. Okay. Alright, just to share you for fish necropsy, or you can apply this to any other organism. Okay, so making observations. Firstly, you just look without making any manipulation. Alright, good legion observations are descriptive and systematic. Okay, you can use this guide, the size, the number, the location, the presentation, the shape. Alright. If you want to take a sample for histopathology, if you want to be more descriptive, place a ruler or any type of measurement near the fish and take a photo. So later on, if you want to look back at the fish photo, right, you need more description. You can always look back and make measurement. All right, I usually make measurement by a software, so it's really easy. And then yeah, it's easy, you know, if you have the photo, you can always look back. All right, the number, how many of it, all right, if it's an ulcer, all right, how many lesions or ulcer are there, right, location, where on the body, is it on the head, is it on the trunk, is it near the caudal peduncle, is it more dorsal towards the dorsal fin, all right, things like that. The presentation, all right, focal, just one, multifocal, a few spots, or diffuse, okay, and then the shape, round, oval, thread-like, ribbon, or tube-like. Some of these can be subjective, but it is always nice to have a photo. Okay, if you take a few photos, you can always refer to somebody who has to have a fresh eye, all right? Sometimes you may not be able to be descriptive, but you have photos, then you can keep, all right, and look back. Texture, smooth, granular, nodular, cheesy, all right. Color and opacity, transparent, translucent, or is it opaque? All right. Sometimes if there is any infection, it will change the color and the opacity of the organ. Firmness, hard or soft. Well, this is as well descriptive, all right, but it is good for you to write in your report. All right, 3D description. Is it depressed, flat, raised? Right, severity. Usually you can give a score, but if you want to be more descriptive, you can say it as mild, moderate, or severe. All right. Extent usually is a percentage of the lesion on the body. Right. This is as I mentioned here, hard to make an estimate. May differ from one person to another, but maybe you can say 50% or the whole body, or maybe 10% means it's mild. Okay. Some of the things that you can see in fish are this description that I will mention, right? You can describe the major or the primary lesion first, and then you can go on the additional observations, all right? Firstly, you can mention is loss of scales, all right? Reddening or otherwise. All right, does the lesion penetrates beneath the surface of the skin and the scale, right? So is it an ulcer that covers, that is deep, an ulcer that is reaching the dermis area, epidermis area, right? Reddening, red or reddish color, not necessarily a penetrating lesion, all right? Can be just, can describe it just as being red. And ulcer is a penetrating lesion underlying the dermis, okay? Muscle or viscera may be visible. The lecture on Friday, right, Pak Sharif showed a photo whereby the ulcer on the fish eats up the head area until it almost exposes the brain, right? So that is a very severe ulcer, okay, and it is a multifocal, right? Okay, lesion location. So this is why it is important to know the fish anatomy, right? Is it near the head? Is it near the vein, urogenital opening, left or right, right lateral line, example, stuff like that, alright? Mentioned before, lesions can be focal, multifocal, diffuse. Okay. Severity, you can use your grade 1 to 3 or 1 to 5, alright, with 0 being no lesion. But if you use a scale of up to 5, then it offers a greater range of observation, and then it sort of is more descriptive, not descriptive, but more easier to differentiate if somebody else is reading the report. Okay. So focal, right, multifocal or diffuse. All right, these are just examples of description of lesion in fish. All right, these are not my photos, right? I tried to get where we can learn how to describe Okay, so this is a goldfish, Carassius auratus. Right, so it has a focal, okay, or rounded or oval, well circumscribed. Okay, so well circumscribed means the edges, right, can be seen. Deep oval ulcer, right, severity of 5, right, so it mentioned here. So they put in a ruler with the fish and then they can actually give the exact measurement, right, located at the left side mid flank above the pelvic fin. So without you looking at the picture, just with the description, you can imagine how does the lesion or where is it, all right? So that is why we need to be as subjective as possible, because the person doing the necropsy is not the same person probably preparing the report or, making the treatment, right? So the ulcer penetrates through the muscle and reveals wow, this is the rib bone. Okay, you can see it's striated there, and some of the viscera. Okay. The left margin of this ulcer not appear to be reddened, but the posterior margin has a cream-colored section, right? Not reddened. Posterior has a cream-colored section extending, and then the size. So what I'm trying to tell you here is when you are looking at a diseased fish, right, and then you want to describe, you should be as descriptive as possible when you are preparing the report, right, so that other people may be able to read it with clarity, right? This is a Atlantic Mahadon, right? So the problem here is that it does not have a ruler. So it is just an estimation or size, okay? Or maybe the person knows the size, so it puts the size here. So this is not an infectious or disease. So it is a fish with a spinal deformity. So it is mostly a genetic thing, right? The deformity consists of a curvature in the dorsal ventral plane and extend from the middle of the dorsal fin to the caudal fin. Alright, severity ranking 3, moderate, animal was alive, and non-remarkable when observed. This is an example of sort of a non-infectious. Okay, so this is also another picture, alright, in which you can describe here. Right, look at the fins here, right? The fins are frayed, okay, or F-R-A-Y-E-D, okay, frayed fin. So the fin this macam not disintegrate, but it's not normal from a normal caudal fin shape, right? So you can mention the size of the fish, how does the fish look like, all right? Or even you can open this dorsal fin, right, with the forceps, open it and then lay it flat, so you'll be able to see if there are any changes on the dorsal fin besides the caudal fin. That's just an example of this is actually a Flavobacterium infection. Okay, so look at the kidney here, all right? So the kidney has a nephrocalcinosis, or in mammals we call it urolithiasis, all right? So when you open up the fish, these are the things that you can see, okay? Once you have observed the other parts of the organ, most likely the kidney will be the last after you've popped the air or the swim bladder, right? And then you'll be able to see these changes on the kidney. These are just examples of the things that you can see during the necropsy, right? So this is what happens when um, a tank or a pond, right, an electric pole, right, fell into the tank or the pond, right, so it kind of like distributes electricity to the fish, and then the fish can get electric shock too, right? So they kind of like jump out of the water or hit something and then injure themselves. So were exposed to electric shock, right? So this is the spinal fracture with hemorrhage, right? So the cable fell into the tank. So these are very not rare, right? You can find this But if you do not know the history, then perhaps it will be challenging. Okay. So besides looking at the internal organs, all right, you can take gill sample, gill clips or gill sample, and then you can observe the changes by using a forceps and unfolding the gill arch, all right. So this is a PGD is actually a mixture of a infectious and non-infectious diseases. Alright, so you can see white spots right at each of the gills lamella. Okay, so white spots, or we call it clubbing, right? Distal third of the filament. So distal third of the filament. Okay, no, this is a better picture. This is a better picture of clubbing of the gills. Doesn't matter what the diagnosis is, if you are able to describe the lesions properly, all right, it will definitely aid in the diagnosis of the disease. Okay, this is also the kidney. This is bacterial kidney disease. Okay, with showing upon necropsy multiple granulomas. Right, very severe. If you think about even in mammals, cats and dogs, right, this severity, alright, of granulomas in the kidney. Alright, we've looked at the functions of the kidney, right, for apa tu, for apa tu, for blood, right, production of blood, for filtering of waste, and also endocrine. Imagine the damage that this would do to the to the fish, and eventually the fish will die. All right, impact fluid functions. Okay. Last two slides. All right, this is the same white nodules you can find on the gill. This is in bacterial kidney disease as well. So in bacterial infectious diseases, you would see multiple lesions on different areas of the body. Okay, I need 5 minutes break. We will meet again in 5 minutes. Okay, all right. Can I just continue one more slide? All right. Sorry, can I continue?
Yes, doctor.
Yes, doctor.
Okay, just one more slide. All right, so this is this is lesion, right? Multifocal granuloma hepatitis. So this is the liver due to BKD, right? You can see the granulomas, right? Multifocal granulomas on the parenchyma of the liver. So this is some of the things that you can see during necropsy, right? Lastly, there are a few videos that I want you to see. Okay. I will copy this on the chat box. We'll copy this on the chat box for you to see. This and another. Oh, Karishna, this is and another. Oops, my daughter is disturbing. This and another video. So there are 3 videos, and I want you to explore the necropsy manual for your own benefit. All right. Let me just copy uh this here. These are the things that I want you to look at after this presentation by me. Take the time to see these videos. Okay, and then I hope that you learn um, something beneficial today because you will practice all this throughout the class and also rotations. And if you're involved in fish medicine, you will be able, you would need all this to practice. Okay. Do you guys have any questions? Oh, and I forgot to mention that for your assessment, 20% will be a continuous assessment. So I've told Farha that I will look at the, I will look at your posters and also your some of your lab reports to give you marks on that 20%. Okay. Uh, do you have any question for me? I suppose tak ada soalan. Um, I suppose tak ada soalan. Okay, do not forget to look at the 3 videos and I want you to explore this website, necropsy manual, to help you learn about how to perform necropsy better. Alright. So for this session, please remember what I've mentioned here about fresh smear sampling and gill sampling and the different approaches of opening up a fish. You will need them. And if you have time or if you are cutting up or eating fish today, right? You may be able to look at where you need to cut for pithing. Also, if you are eating fish today, if you're helping your parents to prepare, then you'll be able to do your necropsy today. All right. So, with fish, necropsy is not hard because you can get or you can practice yourself by getting or buying fish to eat, right? So I hope everything that I've mentioned today is clear. If you have any other questions, you can always email me. Again, I'm finishing up early. Do you guys have any question about this session or any question about, I do not think you have any question about test, right?
Tak ada, doktor.
Okay.
Doktor,
So, yeah. Take credit.
Prof Sharif punya include ke untuk test 1?
Tak. Tapi kalau I include pun boleh je jawab. Kalau I nak include satu je. Satu soalan je. Tapi I tak masukkan pun lagi. So far, actually, I've already set up the questions. It is already in Putra Blast. It's just that you guys cannot see it. It's hidden. I will unhide it during that day lah. Unhide, but you guys can look at the instructions. But it will only allow the system will only allow you to answer at 5:15 on the date lah. Alright. For Afika, you dah try Putra Blast belum? You dah tengok belum? Supposedly your Putra Blast is similar as to the I buat benda yang sama lah to your Putra Blast and the undergraduates. So besides in Padlet, all the notes are also in Putra Blast. So that's another way to get your notes. Test will be by Putra Blast. Test 2 also by Putra Blast. Final exam will be face to face. So I've got confirmation that final exam will be face to face. I already have the date for your final exam already. So that should be no problem. Okay. After I've closed this, please look at the 3 videos that I've given you. So you have some time, 2 more hours. I've always finished early. I'm scared somebody might, ask me. Okay, never mind. I do not want to waste time. Look at the manual, necropsy manual. It actually shows you a few different species of fish. So ada cod lah, salmon lah, a few other fish, tilapia, uh, turbot pun ada. Okay, sampling method, necropsy method usually tak berbeza. Kecuali ikan yang ada one side tu je kan, yang one side, what you call that fish. Itu kecuali itu. Yeah, practical session again, bring the sampling guide, a printout of fish anatomy if you want, and then I'll give you a handout. Handout that you will fill in, you will have to return to me for grading first, but after that I will return back to you. Okay? I have no other thing to say. I hope you have a good day. I will end the class now. Thank you.
Bye, Doctor.
Okay. Okay, bye. Assalamualaikum.
Thank you, Doctor.
Welcome.